Welcome everyone to your own channel, ADP for Learning. I am your own learning partner cum host, Ashish. So, let's begin our learning journey. Today, we are continuing our learning journey about the Interactive Glossary of Genomic and Genetic Terms. And our motto is 5 terms at a time, keep the learning in the mind. The first term of today is Down syndrome or trisomy 21. Down syndrome, also called trisomy 21, is a genetic condition caused by an error in the process that replicates and then divides up the pairs of chromosomes during cell division, resulting in the inheritance of an extra full or partial copy of chromosome 21 from a parent. This extra chromosomal DNA causes the intellectual disabilities and physical features characteristic of Down syndrome, which vary among individuals. Like all individuals with disabilities, individuals with Down syndrome lead full, autonomous, and enriching lives. From infancy, childhood, adolescence, adulthood, and old age, these individuals have many if not most of the same experiences as everyone else. Individuals with Down syndrome, like most people at some point in their lives, may need assistance with certain tasks. They may also have certain health challenges, being more at risk than the typical individual for certain health conditions, all of which are treatable. These individuals are cherished members of each of their communities, going to college, living independently, owning businesses, getting married, and living their lives as they so choose. The second term for today is duplication. Duplication, as related to genomics, refers to a type of mutation in which one or more copies of a DNA segment, which can be as small as a few bases or as large as a major chromosomal region, is produced. Duplications occur in all organisms. For example, they are especially prominent in plants, although they can also cause genetic diseases in humans. Duplications have been an important mechanism in the evolution of the genomes of humans and other organisms. The type I form of Charcot-Marie-Tooth disease is an example of an inherited human genetic disease that's caused by a gene duplication. Individuals with Charcot-Marie-Tooth disease have damage to their peripheral nerves, resulting in muscle weakness. The most common cause of this disease is duplication of the PMP22 gene, located on chromosome 17. This particular gene duplication results in the production of too much PMP22 protein, which causes breakdown of the protective myelin sheath around the nerve, resulting in nerve damage and muscle weakness. The third term is electrophoresis. Electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on their size and electrical charge. An electric current is used to move the molecules through a gel or other matrix. Pores in the gel or matrix work like a sieve, allowing smaller molecules to move faster than larger molecules. To determine the size of the molecules in a sample, standards of known sizes are separated on the same gel and then compared to the sample. Linnaeus Pauling and colleagues reported using the new technology of electrophoresis in 1946 to show that sickle cell hemoglobin carries more positive charges than the normal version on its surface. Today, electrophoresis is used for so much more across biology and medicine. The fourth interactive term is environmental factors. Environmental factors, as related to genetics, refers to exposures to substances, such as pesticides or industrial waste, where we live or work, behaviors, such as smoking or poor diet, that can increase an individual's risk of disease, or stressful situations, such as racism. Genetic studies often take environmental factors into consideration, as these exposures can increase an individual's risk of genetic damage or disease. Many things in the environment impact our health. This includes pollution in the air we breathe, carcinogens in the food we eat, pesticides, lead, increased computer screen time, you name it. Understanding environmental factors is important for understanding genomics. Environmental factors can lead to genetic damage that causes disease. There are also often gene-environment interactions, or a complex interplay between genes and environment, that underlie the risk and development of disease. Since the end of the Human Genome Project, we have seen dramatic improvement in our ability to measure genetic variation. Accuracy has gone up, costs have gone down. Meanwhile, the environmental factors remain numerous, complex, difficult to standardize, and highly variable over time. A key challenge to understanding the influence of genomics on disease will involve figuring out how to best measure, integrate, and analyze environmental data as part of our genomic studies. The last term for today is epigenetics. Epigenetics, also sometimes called epigenomics, is a field of study focused on changes in DNA that do not involve alterations to the underlying sequence. The DNA letters and the proteins that interact with DNA can have chemical modifications that change the degrees to which genes are turned on and off. Certain epigenetic modifications may be passed on from parent cell to daughter cell during cell division or from one generation to the next. The collection of all epigenetic changes in a genome is called an epigenome. The DNA letters in a gene as well as the proteins that interact with said gene are not bare. Both are decorated with chemical modifications that regulate how, where, and when the gene is turned on and off. Epigenetics, also called epigenomics, studies how certain epigenetic modifications may be passed on from one generation to the next. The collection of all epigenetic changes in a genome is called an epigenome. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.